Hi student, I am Dr. D.S. Thavre. Now I share the screen of today's lecture in front of you. Welcome to the 26th lecture of course number Path Triple Food. Title of this course, Diseases of Field and Horticultural Crops and Their Management. In today's lecture, we will discuss about diseases of the brinjal or eggplants. Okay, so in eggplants or brinjal, the Pomopsis blight and Sclerotium blight. So this is the two blight diseases seen in the uh, brinjal crops by Pomopsis vagens and Sclerotinia sclerotium respectively. So yield is drastically reduced because these two blight uh, pathogen infect to the all the aerial plant parts, including the fruits of the brinjal. So that's why economic losses greatly reduced is near about 30 to 50% when the infection seen on the fruit portion of the brinjal. So that's why these two diseases are the most destructive and common disease seen in the brinjal crop. So see, first of all, Pomopsis blight of brinjal. So this is the severe disease seen in the tropical as well as the subtropical areas of the world. So in particularly India, this Pomopsis blight disease was first reported in 1935 in Gujarat. Okay, so this pathogen attacked the leaf, foliage, and also the fruits. But this is the fruit rot phase is the most destructive phase than the other type of the symptoms. Okay, so we go to the symptoms. Okay, so this pathogen develop the typical symptoms such as the damping of then leaf spot and fruit rot. So out of the three phases, the fruit rot phase is the most destructive phase of this disease than the other damping on or a leaf spot pathogens. Okay, so now we discuss about the damping of symptoms and leaf spot symptoms. Okay, so this Pomopsis vagens develop this sclerotia. Means after harvest, these sclerotial bodies may be survival in the infected plant parts. Means under favorable condition, this sclerotia become uh, active and directly infect to the younger seedlings seen in the nursery bed areas. So that's why initially the somewhat light brown to dark brown spots seen on the cotyledons as well as the younger stem of these seedlings. So that's why this pathogen girdle around to the younger stems. So that's why these stems generally collapse. Okay, means this infection generally spread to the upward and downward direction from the collar portion or point of the infection. So that's where these younger seedlings generally fall down and totally dries. Okay, so this type of the infection is known as the damping of symptoms. Then these infections also be developed on the well growing brinjal plants. So that's why initial infection seen on the leaf portions. Okay, means initially brown oval to round spots generally seen irregularly on the surface of the leaf. Okay, then this leaf means this leaf having the small spots grows irregularly manner and cover the entire surface of the leaf. Means majority portion of the leaf showing such uh, oval to round brownish spots. Okay, so these spots grows in the concentric manners. Okay, so this irregular or somewhat concentric rings is developed on particularly leaf portions. Okay, so you see the single lesions. So center portion become gray in colors. Okay, means initially this brown spots are seen, but these brown spots become gray in color, with surrounding somewhat these yellow margins. But after few days or in the advanced stage of the infections, this pathogen means these brownish spots become gray in color or somewhat black in color. Means actually protein bodies are developed, and this protein body is known as the pycnidium. So pycnidium protein body generally produce from this necrotic spots or necrotic lesions. Okay, so in this way, the majority portion of the leaf get infected and showing the blight appearance. Okay, then this fungus also be develop the symptoms on the some branches or twigs of this brinjal and finally reach to these fruit portions. Okay, so you see these infected fruits. Okay, means initially symptoms or disease appears on the developing green fruits of the brinjals. So that's why initially pale brownish or brownish sunken spot generally seen on the outer surface of these developing fruits. But these spots become enlarges in circular manners, and majority portion of the fruits become infected and showing the uh, dark brown to somewhat black in colors. So this is the blackish concentric sunken lesions is developed on the outer surface of the fruits. Means these pathogens just grows in deeply manner from outside of these fruits. Means this infection finally reach up to the inner tissue of the brinjal fruits. So that's why these fruits to be uh, discolored. Suppose if this pathogen enters the fruits through these calyx portions, so whole fruits become mummified. Means after infection of this pathogen through this calyx, so these fruits is not properly developed. So that's why these mummified fruits generally seen or remain attached on the twigs or branches of this is the brinjal plants. Okay, so this is the another symptom seen on the fruits, that is the mummified dry fruits seen or remain attached, may be seen on the veins or branches of these brinjal plants. Okay, so this is uh, symptom is known as the fruit rot. Means finally these infected fruits become rotted because this dry rot and somewhat soft rot symptoms is seen on the internal as well as the outside of these fruits. Means these fruits is totally rotted. Okay, so this is the systematically infection generally spread from leaf to the stem to the fruits, etc. Okay, then causal organism, that is a Phomopsis vagens, or perfect stage of this Phomopsis vagens, that is a diaphoretic vagens. Okay, means these Phomopsis vagens develop the pycnidium fruiting bodies. Okay, so these pycnidia are globules or irregular brown to black found in the infected tissues. But this conidiophore is also developed, and at the tip of the conidiophore, the conidia generally produce. So you see this life cycle or disease cycle of this Phomopsis vagens. Means suppose this damping of symptoms seen in the nursery bed areas. due to the sclerotia or fungus may be survival for long years or long days in particularly disease in the plant debris in the soil. Means this pathogen become active and directly attack to this germinating seedlings in the nursery bed area. So that's why this nursery seedlings get collapsed and finally wilted. Okay, or this fungus also be means this pathogen also be developed to the mature brinjal plant. So that's why these somewhat oval to round brownish spots seen on the leaf portions. Okay, so that's why these pathogens become multiplied within this infected mature plants. So that's why these infections seen on the leaf portion or seen on the wings or stem portion and seen on these developing fruits. After harvest, suppose this infected leaf or fruits having the pycnidium is already produced. So these pycnidium fruiting bodies containing the pycnidiospores, and these pycnidiospores generally produced from these infected lesions and spread from one plant to the another plant through wind or air currents. Means directly these pycnidiospores uh, disperse from these infected tissues and directly fallen on these healthy one host tissues. So these healthy one host tissue become infected. Means in this way, secondary infection is always takes place through these pycnidiospores formed or produced in this infected leaf or infected fruits, etc. Okay, so in this way, secondary infection takes place. Then primary infection, we suppose these infected fruits having the seeds and such seeds may be used for the planting purpose. So 100% this is the produce. Now, or this infected plant part deposits in the soil. So that's why this sclerotial bodies survival in the infected plant parts. Okay, so in this way, primary infection. That's primary infection means use of the infected seeds and infected plant part deposits in the soils. Okay, then last one, that is control measures. So use healthy seeds collected from the healthy fruits and healthy fields. Then collection and destruction of the diseased plant debris properly. Then crop rotation done at least 3 years is required to kill the spores present in the soils, that is the sclerotia. Then seeds are treated with the hot water having the temperature 50 degrees Celsius for 30 minutes. So in this way, you kill the mycelia present inside the seed and seed pod. Then spraying the crop with Zineb 0.2% or mancozeb 0.2% or captain 0.2%. So this fungicide generally applied at an interval of the 7 to 10 days in the nursery areas as well as the in the fields. Then you can also use the the biological agent, that is the Pseudomonas fluorescens and Trichoderma harjanea. So this powder generally applied to the seeds, and this pickets is generally used for planting in the nursery bed area. Or this powder is also used as a foliar sprays at the rate of 10 gram per liters. So make the final volume of this spray solution, and this spray solution generally spray over this brinjal fields. So in this way, you can avoid or eradicate the Pomopsis vagens pathogen present in the brinjals. Okay. Then second most important disease, that is sclerotium blight. So this is the another serious disease seen in the brinjal, appears in the stem blight as well as the fruit rot, causing the yield losses is up to 60 to the 70%. Means damage of the brinjal is mainly takes place by forming the partially and complete wilting type of symptoms. Okay, so this is the symptoms developed in the brinjal plants and foliage as well as the fruit rot, means on the fruits. Okay, means this infection can occur on the any parts of the leaf, foliage, stems, and also the branches, or sometimes this infection also be seen on the fruits. Okay, means this disease seen on the stem portion. So that's why these lesions generally grows in the upward and downward direction. So finally, these pathogen girdle these infected stems. Means initially these brownish lesions is generally seen on the collar portion or stem portion. So these brownish lesions girdle around the infected stems. So that's why these infections spread in the upward direction and downward directions. Okay, so finally these infected tissues become necrotic from these infected portions seen on the stem portions. Okay, so in this way, these plants is totally wilted because these pathogen already girdle around the stem portion. So that's why these above parts of the infected portion or this collar portion to be wilted. Okay, suppose you can seen this stem portion or branches portion. So somewhat this PT tissue is also infected and by producing these sclerotia bodies. Okay, means fungal sclerotia may be seen on the outer as well as the inner surface of the infected stem portions. Okay, means suppose you take the cross section of these infected stems, so some of the sclerotia and whitish mats or whitish mycelia seen on the inner as well as the outer surface of these stems as well as the root surface. Okay, means these brownish sclerotia generally produce on the surface of the infected stem and root surface. Sclerotia means this is the hardish, compact, um, blackish, sorry, this brownish to blackish mustard size sclerotia. This is sclerotia means modification of mycelia under the unfavorable condition to withstand the that condition. So that's why these infected stem and infected roots is finally rotted. Okay, means on rotted surface of the stem and roots, sclerotia and somewhat these whitish mats are seen. So that's why this disease is known as the sclerotium rots. Okay, so this is the infection seen mainly on the stem portion than the fruit. Okay, so this is the brownish sclerotia seen in these petri plates. Means these infected stems isolate and cultured on this artificial media. So that's why this sclerotia is formed on this loose whitish puffy growth. Okay, then causal organism, that is sclerotinia sclerotium. Okay, so this sclerotinia sclerotium fungus reproduce under the uh, favorable conditions. So finally develop this sclerotia, and this sclerotia generally hyaline branch and septate hyper are produced. Means this blackish sclerotia generally elliptical or somewhat is spherical to irregular in shape and generally formed within 4 days of the inoculation on the artificial media in the laboratory under the 25 or ordinary room temperature. So these sclerotia are silvery white seen at the initial growth of this development, but these whitish sclerotia become dark brown in colors after the aging of these cultures. Okay, means primary infection takes place through the infected plant part debris in the soil, means the infected roots and stems debris in the soil. So that's where primary infection initiate from this source. Then ascospores generally produce after the germination of the sclerotia. Okay, then this sclerotia already developed outside or inside the stem and root surface. So these sclerotia and these whitish mats spread from one place to another place through irrigation waters or somewhat is moisture present on the surface of the stems and root surface. Okay, so this is the disease cycle of the sclerotinia sclerotium. Okay, so this is the whitish mass seen on the stem portions. So finally, these sclerotial bodies are seen, and this sclerotia means after harvest, this sclerotia remain debris in the soil. Means so this fungus overwinters as a sclerotia in the plant debris. So these, after germination of this sclerotia, finally develop the apothecium fruiting bodies. So inside the apothecium fruiting bodies, ascus and ascospores are produced, and this ascospore directly fallen on the healthy one host tissue may be seen on the stem surface. So this stem become infected and showing the blackish, sorry, these are brownish regions seen on the collar portions. So this collar portion become infect and finally develop the sclerotia. Okay. Then managements, that is the crop debris should be collected and burned after the crop harvest. Then summer free flowing practices should be done um, to avoid the or to eradicate this sclerotia present in the soil. Then rotation of the cropping pattern with the crops, for example, growing the beetroot, onion, maize, and paddy after the Brinjal crops. So in this way, you can eliminate the fungal inoculum in this case. Then infection generally seen on the stem portion. So immediately you can spray the any fungicide, that is a mancozeb, 0.25 gram per liters, or carbendazim or 2 gram per liters. So in this way you can control these sclerotia, or to control the secondary spread of the disease through sclerotia by application of these fungicides. Okay, so in this way you can avoid these sclerotinia, sclerosium pathogen infect to the brinjal and produce the sclerosium blight. Okay, so in today's lecture, we already discussed about the Pomopsis blight and sclerosium blight disease. Okay, now I end the today's lecture. Thank you.